Hello, everybody. Welcome to a new episode of the Dissenter. I'm your host, Ricardo Lopes, and today I'm joined by Dr. David Haig. He is an evolutionary biologist, geneticist, and professor in Harvard University's Department of Organismic and Evolutionary Biology. He is interested in the intragenomic conflict, genomic imprinting, and parent-offspring conflict. His major contribution to the field of evolutionary theory is the kinship theory of genomic imprinting. He is the author of several books, including the most recent one, From Darwin to Derrida, Selfish Gene, Social Selves, and the Meanings of Life. So, Dr. Haig, thank you a lot for taking the time to come on the show. It's an immense pleasure to everyone.
Thank you. I
Okay.
hope I can be coherent.
Well, you will certainly be. Okay, so let's start with a simple question. What is a gene?
A gene means many different things in different fields, and that's part of the problem in arguments in evolutionary theory. A gene was initially an entity, a hypothetical thing that was constructed to explain a difference in, say, eye color. And so there was some people had blue eyes, some had brown eyes, and the thing responsible for that difference was a hypothetical gene. And in terms of natural selection, there can only be natural selection when there's a difference, choosing one thing or another. So a gene was initially defined as a difference that made a difference, which is Gregory Bateson's phrase also for a piece of information. It was something inherited that made some difference in the organism. In the molecular biology revolution, that definition of the gene has been replaced by a gene as a thing, a stretch of DNA. In one of the definitions, it's a stretch of DNA that encodes for some protein. And it's quite a different concept of gene. I switch from one concept to the other in my own work all the time, but there's a lot of people talking past each other and having different definitions of genes.
Mhm. And that second definition, I mean, the material one, let's say, where you associate one gene with one protein, is that really correct? I mean, because nowadays we also have transcriptomics, and so, I mean, is it the case that really one gene produces one single protein, or the transformations that the proteins undergo afterward, or even the messenger RNA and other molecules in the cell? I mean, is
Yeah.
that still the case?
I think that there's a revolution going on in molecular biology. The one gene, one enzyme hypothesis goes back to studies in yeast a long, long time ago. And then the emphasis was on proteins as enzymes, as catalysts that do things. But we're becoming to realize that a heck of a lot of the control that takes place in the cell is at the nucleic acid RNA level. So there's one gene I've been looking at recently, the IGF-1 receptor. The messenger RNA is about 7 kilobases long, and only 3 kilobases of that is coding for the protein. And the other RNA stuff there, I think, is an information processing device that is deciding when the protein's to be expressed, when it's not to be expressed. And so I suspect that we've been missing a heck of a lot of the evolutionary control that's taking place in what has been called non-coding sequences that aren't specifying protein. The evolutionary concept of the gene, which goes back to George Williams and Richard Dawkins, is just a stretch of DNA sufficiently short to be rarely broken up by recombination, so it acts over evolutionary time as a single unit. And that evolutionary gene could be bigger or smaller than the amount of DNA that's encoding protein. But that's the unit that is subject to natural selection. So once again, we're coming to different concepts of genes for different uses.
Yes, this is very complex, I think. And since you mentioned unit, what is the unit of selection in evolution? I mean, is there one single unit of selection, and would it be the gene or not, or something else?
As usual, people mean very different things by unit of selection. For me, the unit of selection is the difference between two individuals that makes a difference in one survives and the other doesn't. It's the unit of choice of natural selection. There can only be a choice. There can only be selection if there is some difference. But other people have different units of selection. In hierarchical selection theory, you have the gene can be a unit of a selection, or the organism, or the group, but they're actually defining their terms in slightly different ways. So when people talk about the gene as being the lowest level in a hierarchy of units, their concept of the gene is more closely related to an actual DNA molecule within a cell, a material gene. But when Richard Dawkins talks about the selfish gene that is helping its copies in other individuals, it's a piece of information that is present at multiple levels of that hierarchy. And so part of the endless debates and arguments in evolutionary theory is just people using different definitions of genes or different definitions of what they mean by a unit of selection. And that's really why, my recent book, Darwin to Derrida, aims at trying to unravel some of these philosophical issues that most biologists have implicit philosophies of biology, but they often don't analyze the assumptions sitting there.
And does this connect in any way with multi-level selection theory? I mean, is it that when we refer to several different levels of selection, each of those levels has a unit associated with it, or not?
Yeah, you can define a unit as a group or an individual organism. That I would think of them in Richard Dawkins' terms. They're different levels of vehicles that are carrying the bit of information is the gene. My definition of gene, which is that immortal bit of information, that can be distributed across multiple levels of that hierarchy. So a gene in me is present in my sons. It's the same gene that they've inherited from me. So that's another example of just different definitions of genes.
Mm-hmm. And what about group selection? Do you think that group selection occurs at the, I mean, not at the genetic level, let me rephrase the question, but do you think that genetic group selection is a thing? Because there are people out there that don't agree that group selection is a phenomenon that occurs in natural selection, but rather in cultural selection, and so there would be cultural group selection and not genetic, let's say, group selection. So what's your take on that?
Those models are valid models. So I have no problem with group selection models, and I think it's a productive way of looking at things, hierarchical selection. But but I also think a gene-centered view is actually is also highly productive. It's when you develop a theoretical model, what the model does defines what you mean by the terms that go into the models. And so gene selectionists are defining genes in slightly different ways from hierarchical selectionists or group selectionists. And it's not a matter of one set of models being right and the other wrong. They're just formulating the problem in slightly different ways. I think one thing to say about the whole of theoretical biology is that biology is incredibly complicated, and all modeling approaches simplify the underlying biology to become tractable, and so they make simplifying assumptions. And a lot of arguments within theory is just about which simplifying assumptions are we prepared to make in our models. Mm-hmm. But that's a statement about the limitations of us as theoreticians. It's not about a statement about the nature of reality.
Mm-hmm. Yeah. So let's get into some bits of your work in genetics. So I think that when people think about evolution by natural selection and conflict, they usually think about conflict between different individuals to get access to resources or things like that. But you studied intragenomic conflict. Could you tell us about that?
Yeah. So I'm interested. So the assumption of biology for a long time is that an organism is something like a fitness maximizing machine. Our brain is like a fitness maximizing computer, and all of the genes within the individual are working together for some common good. And for most situations, that's a fairly good model of how natural selection is acting. All the genes in the organism are in the same boat, so to speak, and so survival of that organism is in all of their interest. But there come situations where that symmetry of interests among the genes in the genome breaks down, and then that can lead to conflicts, because what enhances the fitness of one set of genes may be different from that of a different set. A very simple example is that genes in the nucleus are transmitted by both males and females, whereas genes in the mitochondria are only transmitted by females. So the production of sons for mitochondrial genes is a dead end. They're not going to transmit those genes further, whereas for nuclear genes, sons and daughters are of equal value. And so this in plants has led to mitochondrial nuclear conflicts many times, where mitochondrial genes cause what is called cytoplasmic male sterility, where they cause the failure of the male parts of the flower to function. Those parts are not transmitting mitochondrial genes. And so that's enhancing mitochondrial fitness, but the nuclear genes are losing all of the fitness that they would get through male function. So that's a simple example of a conflict between different sets of genes within the organism.
Mm-hmm. And that is conflict between the nuclear and the mitochondrial genes, but is there also conflict between different sets of genes in the nucleus, for example?
So that's been part of my career for many years. So at first sight, it seems difficult to see how nuclear genes could be in conflict, because any gene in the nucleus is transmitted, has a 50% chance of being transmitted to every offspring, and that's a symmetrical relation for all genes in the in the genome, and so it would seem to be that the best that they can do is maximizing survival and reproduction of the individual organism. There are a number of places that assumption breaks down. A simple example is transposable elements. A transposable element is a bit of DNA that can make a copy of itself and insert it somewhere else in the genome. And so an ordinary nuclear gene is only getting into 50% of sperm cells or 50% of egg cells, whereas a transposable element, if it makes a copy of itself, inserts it somewhere new, can get into more than its fair share of eggs or sperm. And that transposition comes at a cost of, it increases the rates of mutation and things like that, so that there is a conflict within germ cells between transposable elements attempting to make extra copies of themselves and law-abiding Mendelian genes trying to shut down transposition. Mm-hmm. Now, interestingly, those two sets of genes in most of the body, cells of the body, which don't produce gametes, have symmetrical interest. Transposition in my body can increase the chance of cancer, but it doesn't enhance long-term transmission of the transposable element. So I think transposable elements in the rest of the genome are cooperating in the somatic cells of the body because they have a mutual interest in that somatic body reproducing, and the conflict is focused then within the germline cells.
Mm-hmm. So it occurs mostly at the level of the germline cells. So, and even between cells of different parents, I mean, isn't there also what you call, I guess, parental antagonism, and that is conflict between the genes coming from the father and the genes coming from the mother, and in terms of expression of one or the other and things like that?
Yeah, so that is goes back to my PhD thesis back in the 1980s. And so once again, I have at each locus on my chromosomes, I have a gene I get from my mother and a gene I get from my father, and they're equally likely to go to my children and to my grandchildren, my great-grandchildren. And so they have a symmetry of interests there. Where that symmetry breaks down is if a gene has effect on non-descendant relatives, not my offspring or my grand-offspring. And the simplest example is thinking about fetal development. The maternal gene in the fetus is definitely present in the mother and has a 50% chance of being transmitted to each of the mother's other offspring, whereas the paternally derived gene in any particular fetus is absent from the mother. And the only chance it has of being in the mother's other offspring is if she has extra children with the same father. And so now we've broken that symmetry. The paternal gene in the fetus is less likely to be present in the mother's other offspring than is the maternally derived gene. And so paternal genes are selected to maximize, not to maximize, but to increase costs on the mother for benefits of the fetus because they're less likely to be present in the mother's other offspring. And so this has led to the evolution of a phenomenon called genomic imprinting, where some genes are doing one thing, being expressed, say, when they come from the father, but not when they come from the mother, or the opposite way around. And from the point of view of thinking of a gene as a bit of information that continues across generations, a gene is playing a conditional strategy, make the placenta grow larger when I come from a male, but reduce placental demands when I come from a mother. And so it's a conditional strategic behavior. And this is phenotypically played out then in a conflict within the organism between genes it gets from its mother and from its father. So there's good evidence that in placental development, genes of paternal origin are favouring greater growth of the placenta, and that is being inhibited by genes of maternal origin. But these sort of conflicts can be expressed after birth. One that I think is particularly fascinating, when you talk to new mothers, the thing that they complain about all the time is that they feel exhausted and that they're not getting enough sleep. And this is because the baby is waking them up to feed at all times of the night. We think, sure, babies need food, but a rabbit, for example, a baby rabbit only suckles once every 24 hours. The constant waking of human babies, I think, is partly an adaptation of the baby to tire out the mother, because that delays the time till the mother returns to fertility to have her next child. And so in this conflict, genes of paternal origin are favouring a longer delay to the next child than our genes of maternal origin within the child. And so I've been particularly interested in two disorders of genomic imprinting called Prader-Willi syndrome and Angelman syndrome. And a Prader-Willi syndrome child is lacking genes of paternal origin, and those children don't suck, so they need to have a tube put into their stomach to get enough food, and they sleep through the night. And that's saying that the paternal genes that are missing are promoting suckling, which is extracting resources from the mother, and that they are tending to wake the child up, which is tiring the mother out. Angelman syndrome is the same set of genes involved, but in this case, they're missing genes of maternal origin. And those babies some of them reported to be awake 21 hours out of 24. And so the missing maternal genes are putting the child to sleep. When you have a deletion of those genes, you've got a child that's awake all the time. And so that's an example now of a conflict between maternal and paternal genes within the child about whether it is to demand more food or whether it is to take a nap.
Mm-hmm. And that's interesting, because even though there's that conflict between those genes from the two different origins, the maternal and the paternal side of things, when they are together in the same organism, I mean, they lead to those results that are important for the organism itself, like, for example, suckling or sleeping well and things like that, right?
Yeah. So it's so genes within the organism have both conflicting interests and common interests. It's like any society. And so they have common interests in the well-being of the particular body that they're sitting in, but they also have potentially divergent interests in the well-being of other bodies. So in terms of relatives, most of our non-descendant relatives, if we leave aside our children and our grandchildren, our cousins are either our mother's relatives or our father's relatives, but if we're not inbred, they're not going to be both. And so my maternal genes have an interest in my maternal aunt's children, but my paternal genes don't have an interest in the maternal side of the family, and vice versa. And so I think that genomic imprinting, these conflicts between maternal and paternal genes, will play out in all sorts of social behaviors within the family, because a relative is an individual who's share some genes, but not all genes, and that similarity but difference creates the potential for these social conflicts among genes.
And there's also parent-offspring conflict, correct? You already mentioned some examples of mostly maternal offspring conflict, but there's also paternal offspring conflict, and so sometimes the interests of the offspring are not well aligned with the interests of their parents, for example.
Yeah, that the parents gain fitness, value all of their offspring at first sight equally. Each of your offspring has the same chance of inheriting parental genes, but once a gene finds itself in an offspring, it's definitely present in that offspring, and it's only got some chance of being present in its siblings. So offspring tend to value themselves more highly than their siblings, whereas parents tend to favor more equitable distributions of resources among siblings. And so the theory of parent-offspring conflict is closely related to competition among siblings for resources, but in this case it's competition among those siblings for resources coming from their parents.
Mm-hmm. Right. And so let me ask you another thing, because we've been talking about genes. We know nowadays that genes are expressed differently in different environments, and we have disciplines like epigenetics. So what is the importance of that for the things that we're talking about, that genes are differently expressed in different environments?
It's genes playing conditional strategies. The best thing to do differs in different environments, and so they evolve conditional behavior. Just as within my body, some cells, some copies of genes are ending up in liver cells and others are ending up in muscle cells. And so in those different environments within the body, muscle-specific genes are expressed in muscle but not in liver. That's a simple example, everyday example of genes doing conditional strategies. Think of I and my complexion. I don't tan, but a lot of people in Portugal, if they sit out in the sun during the summer, their skin becomes darker. And in the winter, when there's less sunlight, the skin becomes lighter. And so that involves changes of gene expression within melanocytes, and it's adaptive conditional behavior. Paler skin is better for synthesizing vitamin D, which needs sunlight, but darker skin protects against UV radiation, which can cause skin cancers and sunburn and things like that. And so it depends on the environment, how much sun there is, whether the melanocytes in the skin synthesize melanin or not. Simple conditional behavior. So the genomic imprinting I was talking about, where genes are doing different things depending on which parent they're inherited from, is another example. In this case, it's a past environment that's determining what the gene is doing. If the gene in the last generation was in a female, do one thing, reduce your demands on the mother. If the gene was in a male, the previous generation, it does something different, increases demands on the mother. So I see all of these sort of differential gene expression as just being strategic genetic behaviors, and we tend to think of that strategy that that everything starts again in this generation, so it's only the current environment that genes are responding to. But if we were growing up under, there's a nice example from the Japanese Navy in the 1920s. People of Japanese descent who were born in Okinawa, where the temperature was hot, had more functional sweat glands for cooling off than people who grew up in Tokyo, where conditions were cooler. And those differences persisted into adult life. So here is an example that early events during the environment you were exposed to as a child is having long-term effects, and some of those long-term effects are changes in gene expression.
Mm-hmm. Mm-hmm. So you mentioned development, and there are some changes that occur in gene expression according to the ecological and or environmental conditions people or other animals or beings are exposed to. Is there any conflict between gene-selectionism and developmental theories, like, for example, developmental systems theory? And could you tell us about that? What is developmental systems theory about?
So developmental systems theory, it's people who feel that in understanding development that that too much importance is given to genes, and from their perspective, genes are just one of the elements that takes place in development. So they're really concerned with mechanisms. What are the mechanisms of development? And so genes are responding to the environment. And so the environment is having a causal role in development. And so those people, dislike, for example, Richard Dawkins and his selfish gene-selectionism. But this is another example where people are actually using different definitions of genes. The developmental systems theory is concerned as the causal mechanisms by which a gene as a material object is causing development to take place within a generation. And so it's just one of the materials that takes place in development. And the food you eat also has a role in that developmental process. Whereas Richard Dawkins is talking about how does that, the gene is a piece of information that is changing over time through natural selection. And there isn't an equivalent in the environment, I would argue, that is retaining a memory of past selection and being transmitted from one generation to the next. We do change our environment, but there isn't a textual record of those past environments, like there is a textual record of past genes that have survived past selection. Now, from the evolutionary perspective, where does the information in genes come from? The information in genes comes from the environment that selects. So some genes survive and reproduce better than others, and what's doing the selection, what's putting the information into the genes is the environment. So as a gene selectionist, it's the environment that is choosing which genes we get to see in future generations.
Mm-hmm. And are there any elements here about all that we've been talking about that are Lamarckian in any way instead of Darwinian? I mean, for example, epigenetics, aren't there at least some species where people have proven that there are elements that get transmitted differently according to the environment the parents, for example, are exposed to?
Yes, there are some examples of transgenerational inheritance of that sort. And once again, genomic imprinting is an example of that a gene that was in a male body is having an imprint in the next generation that it was in a past environment, a male body, and that changes its behavior in the next generation. So there is no obstacle to those sorts of transgenerational effects. But imprinting is an important one, that there's no long-term heritability there, because the gene that I get from my mother, which has the maternal inherited imprint sitting on it by epigenetics, when I pass that gene on to my children, they're going to have the paternal imprint sitting on it. So the gene does carry environment from past environments, but that can be erased and reset. And so I think a lot of talk about Lamarckian epigenetic inheritance is confusing long-term evolutionary change with these short-term strategic adaptive behaviors of genes. Now, people make a big thing about inheritance, for example, of methylation differences sitting on the DNA sequence, and so they're saying that that's non-Darwinian. That doesn't make any sense at all. The basic Darwinian theory in population genetics was figured out before we knew that genes were made of DNA, and so Darwinian natural selection applies to methylation differences just as well as it does to sequence differences. And so this is another example of, so from the Darwinian selectionist, the gene that's being subject to selection, it doesn't matter whether the difference that you're looking at is a methylation difference or a DNA sequence difference. It is subject to Darwinian natural selection. It's not a Lamarckian process. But if you think of a gene defined as a bit of DNA with a particular sequence, then methylation seems to be something other than genetics.
Mm-hmm. So let me ask you a general question. Do you think that there's any teleology in natural selection? I mean, because at least for lay people, when they hear about natural selection and the function that different adaptations, for example, have, and because sometimes we look at things linearly, it seems that there's a purpose in natural selection, that things follow a certain way and they improve over time. But is there something of that in natural selection or not?
So the way I put it is natural selection is a process without purpose, without direction. It's just the environment selecting among variants, but it produces beings that have purposes and intentions. So historically, so there are two concepts of teleology here. One is just that living things are doing things for reasons. That when your skin tans, it's to protect your skin from ultraviolet radiation. That's a teleological process. Now, historically, the only way that people thought purposes could arise were that they arose in the mind of some being. And so they argued from purposefulness in nature to some designer, some creator, some divine mind that came up with, that designed the organisms. What I think Darwin did was he said that you can have the evolution of purposive behavior, beings that behave teleologically, do things for reason, without there being some overall directing purpose or goal for the process. So the way I put it is natural selection doesn't have purposes or goals. It's not directed to any particular outcome, but it produces organisms that have all sorts of purposes and reasons. And I think that's the really radical thing that Darwin did, was that he showed how the world of meaning and purpose could arise through natural selection in a world that was without purpose and without meaning. And so that's part of what my recent book is about, because that's a minority perception of what Darwin did. So the dominant explanation is that he showed that there was no purpose in the world, that he rejected the argument from design. I think that's influenced by turning to physics. So physics rejected teleology a long time ago because physical things, they're not subject to natural selection and they don't have purposes. But living things, because of natural selection, things that worked in the past get to do the same things in the future. So the reason why you have genes that allow you to tan is because tanning in the past favored the reproduction of those genes for that behavior. So once again, this is the example how the information is coming from the environment to the gene. So the way I put it is that the effects of genes have a causal role in which genes get to reproduce, so that all of the adaptive things we do, the genes that cause that those behaviors then allow that gene to reproduce or not. So there is a causal pathway coming from the effects of genes in the past to the reproduction of those genes. So that's the teleology I see in the natural world.
Mm-hmm. And that purpose applies to both humans and other living beings, right? Because I mean, it doesn't matter if it is something conscious or not. Right.
Yeah, I leave aside the question of consciousness. In fact, I think consciousness is overrated. And there's quite a bit of evidence even that most of our conscious perception is coming after we've made a decision. And so most of the fascinating things that living things do, and they're doing all sorts of purposeful stuff. So when I climb a hill, my heart starts beating faster to deliver more oxygen to the blood. When I take a meal, my pancreas produces insulin to reduce my blood sugar levels. All of that very purposeful behavior is taking place without us being conscious of it at all. So my general feeling is that in terms of the amazing things that living things do and their amazing feats of interpretation, most of that takes place without consciousness. In my book, I do talk briefly about when do we use consciousness. I think it's used in two sorts of situations. One of them is bringing attention to bear and making plans. The example I use when on a summer day when I'm walking to work, I make all the decisions where I'm turning without thinking about it. I know the way to my office, and I'm not really thinking about it as I go. That's automatic decision making. And the side effect of it is that when I mean to go to the dry cleaner to take my clothes there, if I don't have that in my consciousness, I end up in the office with my dirty laundry sitting in my hands because I forgot that was my plan of what I wanted to do. But when I'm walking to work on an icy morning when the street is covered with ice, then I'm paying very, very close attention to where I'm putting my feet. And so I'm using visual, all sorts of different bits of information because there's something, attention is needing to pay to it, to that problem. So that's one thing that we bring consciousness to bear. Another really important thing is in justifying ourselves to others. So if I tread on your toe, I apologize. I say I didn't mean to do that. It was an accident. And so that's one thing that we clearly use self-consciousness for, is explaining our reasons to other individuals so that they can understand us. And, there's quite a lot of evidence that sometimes there's elements of self-deception or deception in the way we present ourselves to others. So I think consciousness is definitely interesting, but in biology, it's just a special purpose adaptation used for specific sorts of problems, and most of what we do without consciousness.
Mm-hmm. Very well. Okay, and my last question, and still focusing on your last book, From Darwin to Derrida, is what is meaning, and how do we get at meaning, or at least at creatures that attribute meaning to things, to themselves, to the world around them through natural selection?
So the answer I give in the book is that the meaning of information is how you interpret the information. Meaning is the choice. You take in information from the environment or from other individuals, and the meaning of that information for you is what action that you take. And so I synonymize meaning with the use of information in choice. And so that's attempt to cut out a lot of, there are endless arguments about where does the meaning reside in the information? And I'm saying it's not in the information. The meaning is in the interpretation of the information.
Mm-hmm.
And yeah, go on.
Yeah, and in terms of, let me just interrupt you there just to present a follow-up question. So if meaning has to do with how the specific organism interprets the information and the action that it produces in response to that information, does it mean that other animals, for example, also have meaning?
Yeah. So when a growing plant chip grows towards the light, the meaning of it perceives the light coming from some direction, and the meaning for the plant of that light is, this is something to grow towards. It's the action of growth. Then I would say is the meaning of that stimulus to the plant, and so that's why Derrida is sitting in the title of the book. The argument is that the meaning of information is whatever an interpreter interprets it to mean. And so two people can read the same book and it means different things to them. And it's not an issue of one of them being right and the other being wrong. It's just that they're interpreting the same information in different ways. So at that level, I'm sort of a relativist. The meaning of any bit of information is whatever you interpret it to mean. That doesn't mean that all interpretations are equally valuable. Some interpretations allow you to survive in a harsh world, and other interpretations are wrong in the sense that you make the wrong decision and you were hit by a car or whatever. So one criterion of choice among interpretations is their usefulness in life. Some interpretations are more popular than others. Some interpretations get taken up and are transmitted from person to person, whereas others are unpopular. Those are different criterion of success there. So one can choose among different interpretations, but the meaning is in the interpretation, not in the information received. And what organisms are, we are interpretive devices. So life, in a sense, is interpreting information about the environment to make meaningful adaptive choices in the world.
Mm-hmm. Yes, that's a very interesting perspective. So would you agree then that because different organisms evolve in different ways, I mean, the way they perceive the world, for example, is different from a human to a dog to a plant, let's say, if we can even talk about perception in a plant, I'm not sure, then through natural selection, at least pragmatically speaking, we get closer, or we get an approximation to what reality really is or what the world is, because there are interpretations that work better than others.
Phenomena, the things that we perceive, they are interpretations of the world. So the meaning of the input to our eyes, the interpretation is the perception that we get. There's a lot of evidence that, the colors that we see when we look out at the world, they're not out there in the world. They're our interpretations. They're symbolic interpretations in the mind. And different organisms have different color sensors. So Cassie Stoddard has recent beautiful work on hummingbirds, which rather than having 3 color receptors, have 4 color receptors. So they can make distinctions of color in the world that we cannot make. And if something had 5 color receptors, they could see things that hummingbirds couldn't do. So the perception of our perceptions of the world are a form of virtual reality. They're a model of the world that we construct to make sense of the world. And I just wanted to put in a word for plants. Of course, plants are perceiving the world all the time. Part of the problem is that they respond to the world at a slower pace than we do. So that if you look at time-lapse photography of plants growing, they're clearly, they show a lot of behavior and are purposeful, but it's just at a slower rate than we do. And that's because they're stuck in the ground. They're not able to move rapidly. So their meaningful behavior is taking place over longer time scales than our meaningful behavior does. And so we tend to think, oh, they're not perceiving much. But the plants outside my window, they can clearly see the world in some sense. They have photoreceptors. There are branches that know when to stop. That there's one tree that doesn't grow into the wall. So somehow it's perceiving the wall there and knowing when to stop. So I think plants, I started off as a botanist. I think that they don't have centralized nervous system like we do, but I think they are actually perceiving quite a lot of the world and responding adaptively to what they perceive. And I haven't even started to talk about what's happening underground. Their roots are living in a very complicated environment, but it's out of sight and out of mind. We're really not seeing what those roots are doing. That's one of the great undiscovered frontiers in biology of what is happening under the surface of the soil.
Yeah. So this will be really my last question. So, in terms of interpretations, as you mentioned, do you think that, okay, so for example, in science, we are trying to understand what the world is and how it works? I mean, we're trying to arrive at objective truth, let's say, but since different beings, living beings, have different, let's say, interpretations of the world, and you mentioned that, for example, the way we perceive the world is sort of a virtual reality. Would you agree then, or do you think that we can talk about a sort of pragmatic truth, in the sense that if a behavior leads toward an increase in fitness, then that interpretation that particular living being has of the world is more correct than an alternative behavior that doesn't produce the same results?
Yeah, that's the interpretive systems we have evolved give us objective information about the world, because forms of perception, interpretations of the world that gave inaccurate, I shouldn't say inaccurate, but less useful information do not get to survive. So the phrase I use is that our subjective experience is objectively grounded in the need to know. It's the things we need to, our interpretive systems have evolved to provide the information we need about the world. It is a virtual reality, but it's a very, very useful virtual reality. It's particularly, and in general, we can trust our perceptions when we're dealing with the inorganic world, because the inorganic world is not playing games with us and trying to manipulate our interpretations. It's much less clear that we can trust our interpretations in my interaction with you and other people in society, because people play games with evidence. People rationalize their own self-interest all the time. And so the way they say I objectively view the world is often very self-serving because of conflicts among human individuals. So I do believe that we can trust our perceptions of the physical world. I think we should have some skepticism of the way things seem to us in our interaction with other people. We need to look at what are our motives for seeing the world the way we do, and need to accept that other people have different motives and will see the world in a different way than we do.
Mm-hmm. Okay, so Dr. Haig, let's end our conversation on that note. And before we go, I will be leaving links to your books in the description box of the interview. Would you like to mention any places on the internet where people can find your work?
If people want to get in a conversation, just send me an email and I will try and respond and answer when I get the time. Yeah. Mm-hmm. That's the simplest way. Send me a message.
Okay. So again, Dr. Haig, it was a real pleasure to have you on the show, and thank you a lot for taking the time.
Okay, bye, people.
Hello everybody, thank you for watching this interview until the end. As you might have noticed, I've started this channel back in February 2018 and have been putting out regular interviews with academics and intellectuals from a variety of fields. And to keep the channel sustainable, I would like to ask you to please visit my Patreon page and to consider making a pledge there. And I also have links to PayPal in the description box of the interview. Otherwise, and if you like what I'm doing, please share it, leave a like and hit the subscription button. I would also like to give a huge thank you to my patrons and main supporters, Karen Litske and Blanchett, Perga Larsen, Lau Guerrero, Francis Ford, Hans Frederick Sunda, Ricardo Vladimiro, Greg Healy, Adam Kessel, Olaf Alex, Jonathan Wiesel, David Diaz, Anian Kata, Jacob Klimpy, Matthew Whittingbird, Arno Wolf, Tim Hollacy, Enric Alenius, John Connors, Paulina Barron, Philip Force Connolly, Jerry Mueller, Herbert Gintis, Rutger Voss, Bo Weingard, Rebecca Newberger Goldstein, Dan Demetri, Robert Windiger, Rui Inácio, Arthur Coe, Marco Neves, Max Belby, Colin Holbrook, Susan Pinker, Thomas Trumbull, Bernardo Seixas, Pablo Santurbano, Simon Columbus, Jorge Espinha, Phil Kavanagh, Corey Clark, Mark Blythe, Roberto Inguanzo, Michael Stormir, Eric Neumann, Samuel Andreev, Tiago Nunes, Bernard Hugney, Alexander Dunbauer, Omari Hickson, Felicia Stevens, Fergal Cusson, Evan Bodrenko, Hal Herzog, Nuno Machado, Don Ross, João Alves da Silva, Jonathan Leibrant, Oslam Bullitt, Nathan Nguyen, Stanton T, Samuel Correa, Eric Hines, Mark Smith, J.W., João Eira, Tom Hummel, Sardus France, David Sloan Wilson, and Yacila Deza Araújo. My producers is our Weber, Jim Frank, Lucas Stafiniak, Dr. Ian Gilligan, Sergio Quadriano, Luís Caetano, Matthew Lavender, Tom Van Egdam, Curtis Dixon, João Linhares, Benedict Mueller, Vega Gidi, and my executive producers, Michel Rugieski, Rosie, and James Pratt. Thank you for all.